I'm Lynette. I'm from the Genomics lab at Source Bioscience, and I'm just here to give you a little overview about what we offer. So as a company, Source Bioscience has three main divisions. We have our classical healthcare department, which is based typically out of our Nottingham site. That's a lot of the traditional histopathology type work. We also have offices in London and Chichester that support the digital arm of pathology, and we work extensively with both NHS and private hospitals around the UK. As part of that portfolio, we also do offer a few diagnostics and clinical tests, broadening into the precision medicine arena. Our second department is stability storage, and that's typically based out of Rochdale in the UK, but we do have sites around the world in both Ireland and San Diego. So that's typically working with a lot of pharmaceutical companies who are looking for ICH conditions and product testing over time. And then today, obviously, what we're more interested in explaining to you and showcasing is our genomics services. So from Genomics, we are a company that can provide a whole range of different customers with a lot of different services. One of the most important parts of our business as a whole is that we are very much focused on quality. So you can see a range of the different companies that we work with, either following guidelines by the relevant organisations and bodies or being actively accredited according to ISO standards. So we have 9001 ISO accreditation across our business, and within the healthcare fields, we have ISO 15189. We do also get inspected for our good clinical practice according to the MHRA too. As a commercial company that does deal with both data and healthcare information, we also have a big focus on data protection and have a whole range of systems in place to make sure that that's protected first, which we know is quite an important factor to people who are outsourcing their work. So the Genomics Lab is our new centre of excellence. We have a custom design facility that we just moved into at the end of last year. We have essentially the state-of-the-art labs with full range of services to cover the whole portfolio, which include our genomics across NGS, our multi-omic biomarker discovery services, our extraction services, and our Sanger sequencing service. These are just some pictures of our nice shiny new labs. We do occasionally have events where you can come and see them for yourself. So if that is something you're interested in, please definitely keep an eye out for those. One part of our business for this, our genomics is Sanger sequencing. It's something that we've been doing for a long time, and we have a really great relationship with a lot of customers, providing them with a really fast turnaround time. For this service, we offer either overnight with an evening collection, so customers in the Golden Triangle can drop off their samples, say at about 3 o'clock in the afternoon. They'll get to our labs by sort of 6:30, 7 o'clock. We'll process the reactions and we'll put them on our sequencers with data being dispatched overnight. So the customer will literally come into data first thing in the morning for them, so they have no delays to their own workflows. Our main site is in Cambridge, but we do also have a facility in the northwest, so that we can also offer faster turnarounds to our north of England customers too. There are a few additional services we can offer for the Sanger sequencing community, such as plasmid extraction, where you can send your cell pellets ready for both processing and full sequencing, and we can offer extra services such as PCR cleanup of amplicons, and additionally, for those that are able to prepare their own sequencing reactions, we can do the run-only loading onto the 3730s too. Our main focus, and obviously what I'm here to talk to you today, is what comes out of what's turned our NGS lab. So we offer a whole range. We are typically historically Illumina focused, and we have a wide range of instruments to cover all their data needs. We have MiSeq, NextSeq, and NovaSeq. So we can do a lot of different sequencing data output requirements across those platforms using a range of Illumina-based chemistries to cover both your 4-channel and your 2-channel options. We do offer quite a comprehensive run-only service. So for researchers who like to keep certain parts of their process in-house, you can perform all your upfront work and just send it off-site for literally loading on the sequencer. Otherwise, we're happy to perform the whole service. This is just a range of some of our kind of typical routine processes. So we're doing traditional RNA sequencing using poly-A and RNA depletion methods, low input methods. We obviously offer whole exome and whole genome sequencing packages, methylation, and then a whole raft of the newer technologies also to support single cell and spatial transcriptomics. And one of the other times, the biomarker industry that follows more of a traditional NGS application that we support is the Illumina TSO 2C oncology panels. So we are very much looking to be in the world of genomics and to move forward with the technologies. We also want to provide the best service to our customers. So we do, where possible, look to join schemes to be accredited service providers and perform in either testing with the service providers or to do everything that they ask on a level to be recognised as a partner lab with these companies. This is just a few of the recent ones that we work with. So we are a certified provider for the 10x spatial transcriptomics service. So this obviously allows you to take your RNA sequencing level down to linking it to the position of the tissue where those cells are coming from. We offer Oxford Nanopore for your long read sequencing needs, and we can do this using a whole range of flow cells using our GridION and PromethION. So the Nanopore sequencer has a whole range of options too. These are the most common ones, but it is also great for looking at things where you have modified bases, which traditional sequencing may not be able to take on as well, such as also the methylation-seq and for looking at some structural variations. Obviously, we're here today to talk about Olink. So we are offering the Explore platform, and that is the large panel Olink process that will ultimately run on the NovaSeq instrument. I'm not going to say too much right now because you're going to get plenty of information about that. To further our portfolio, we also offer optical genome mapping. So this is a method that's often termed high definition karyotyping, and it allows us to look at the genome in another way. So we can look at that to look at large scale structural variants. And this can be used in its own across a range of topics, or it can be used in comparison and in complementary to our whole genome service offering, so that the whole picture can be looked at of the genome and any events that are occurring there. We offer extraction services. We provide a whole range. We can do smaller projects using a range of sample inputs up front of our NGS technology, or we offer kind of the larger bulk processes using our automated systems, which typically a lot of blood and saliva extractions across DNA and RNA. We have worked with a lot of different things. Most commonly, we're working with basically cells and tissue every day, but we can support upfront of our products such as the TSO by performing ctDNA extraction, FFPE extraction, and we do work with the novel environmental across the metabiome as well. One thing that we like to also be able to prove that we can do, and that would come under the GCP arena, is that obviously we have a lot of experience, skills and equipment for our whole core portfolio that we've just mentioned, and we are able to support trials that are clinically recognised and do come under GCP. So that is definitely something that we can support, and we do have dedicated clinical trials managers as well for that service. So one of the reasons why you may wish to use us is maybe you don't have access to all the technology or the equipment in your own hands. Obviously, some of the instrumentation is quite specific and expensive, so we are able to, for example, provide the run only, but we also do work with other customers who are starting up their idea and want to build new portfolios, new technologies, but in that short term don't necessarily have the lab skills or the lab space or the budget to do all the laboratory work themselves. So we can offer a whole range of services to our customers, and I'm more than happy to provide quite a thorough level of support and technical advice where needed. We like to say that we are one source. We are genomics and multi-omics focused across the whole range of genomics, epigenomics, transcriptomics and proteomics. Obviously, this has just been a really quick run through, but we are very passionate about what we do. So if you have any questions, please feel free to come and talk to myself or my colleagues, Richard, Jake and Laurie at the back there later, and we're more than happy to tell you what else we can offer or to discuss how your project needs might fit into something that we can support too.
Okay, Yeah, so my name is Richard Vivier. I'm a business development specialist at Olink. I have another colleague here, Mariana Fontes. He's from Scientific Affairs. He has a background in neuroscience, but also covers other things. And see our director here, Pavel Godzielewski, is also here. So we'll be available after the meeting, or obviously during this conversation as well, to answer any questions. So I'm going to give a kind of short intro to Olink technology. And my colleague Mariana is going to give sort of more scientific overview with some nice case studies to show how you can actually use the technology. So Olink, Swedish company, we're global. So we have sites in the US, Sweden, and in Asia. We're adopted by, say, all the top pharma. Very scientifically driven company. I joined the company just over a year ago, and that's one of the reasons I joined. In addition to loving the technology, I think it's a fantastic technology. There are around, I think, over 1,300 peer-reviewed publications that have used our technology, which gives you an indication of the utility of it. We have many, I would say, very good KOLs using our platform, different consortia. I think Mariana's going to give an overview of maybe one or two of those as well. So why would people use our technology? You could use it for any particular application where you need a biomarker. In terms of drug development, one of the applications could be to reduce risk during the drug development process, save you time and money. So as I was saying, so one of the reasons people might want to use our technology in terms of drug discovery is to reduce risk in drug development. But a number of applications you can use it for. Identification of the right drug, right dose, the right time to the right patient. Understanding biology, it's used for a number of applications, wellness, maybe you just want to identify novel biomarkers. But actually the number of uses is endless. In terms of, let's say, drug discovery, you can use it for drug discovery or for basic research. There's a number of applications listed there. Translational research, or for drug development. Historically, there have been a number of, say, technological challenges around proteomics, including lack of sensitivity, lack of specificity, high sample consumption. So if you have precious samples and you have a limitation on sample volume, that could be a limitation for you, and also limitation on dynamic range. And the Olink proximity extension assays actually got around all of those hurdles. So we have high sensitivity, typically down in the low picogram per mil range, but we have also some assays that have even better sensitivity than that. Specificity is actually unparalleled. And I'll go into some details later on when I describe how the technology works as to how we achieve that. The sample volume is really, really minimal. The actual assay physically takes 1 microliter, although when you're running a study, they might ask for some extra because there's liquid handling involved, so they need some extra volume for that. And also the dynamic range is very broad compared to some of the other platforms out there on the market. So the plasma proteome is thought to range dynamic range around 10 to the 10, and people consider maybe mass spectrometry as being the gold standard for a long time. I will say that our technology is very complementary to mass spectrometry. This graph is quite a nice graph because it illustrates how basically Olink can complement mass spectrometry, or actually you can use it as an alternative because there are some advantages such as the throughput, for example. And also there's no preparation, sample prep involved for assaying plasma and serum. With mass spec, you typically have to remove the highly abundant proteins such as albumin to give you a chance of measuring things that have lower endogenous levels. So that treatment actually makes it hard to basically replicate your data between different labs, and also it's time consuming and it's altering the matrix. So you're not actually assaying the matrix in its native form, you're performing some kind of manipulation to it. So with Olink, you don't have to do that. Most of our panels, you will just, for plasma and serum, you just assay them as is. For the more abundant proteins, you might have to do a dilution, but there's no preparation typically involved. So for mass spec, you're typically picking up proteins in, you can see where my mouse is pointing, in this kind of range. With Olink, you can pick up everything, including these lower abundant proteins. So in the low picogram per mil range, you're going to pick up things like cytokines, which are molecules that are quite interesting to a lot of researchers. So how does the technology work? So it uses something we call dual antibody recognition. So for every analyte that we measure, we have 2 specific antibodies. And each of those antibodies are conjugated to a specific oligo. You actually need both of those antibodies to bind for those oligos to be in close enough proximity to then hybridize and basically generate a unique barcode for that protein. So if you only had one antibody binding, you won't get hybridization of a complementary oligo, so no signal. If you had the correct antibody binding at one site and maybe a non-specific antibody binding, then the oligos aren't going to be complementary, so they're not going to hybridize, so it means no signal. And once you've got hybridization of those oligos, you then have a unique barcode which you could amplify by PCR, and you can basically detect either using NGS, which we do for Explore, or by qPCR, which we do for, say, lower throughput platforms. So in addition to the assays, I just wanted to highlight, we have other products. So we have the main assays, which I'm going to highlight today, but we also have something called insights as well, which are kind of applications that complement, let's say, the assay side of things. So you get a really complete experience. I think Mariana is going to go into a little bit more detail about insights, but there's basically data in there that you can access for free. We have data analysis tools that are available there for free, and other things, pathway mapping, etc. I'll talk a little bit now into the actual products that we have. So this is the whole portfolio. I'll start off talking about the Explore platform. So this is the platform that Source are able to run for you. This uses an NGS readout, as I mentioned earlier. We have currently Explore 307.2, which consists of a menu of around 3,000 analytes. I say currently, we're planning to expand that to a larger number, hopefully sometime in the near future. So you have the option, I would say, a less biased approach of being able to identify a biomarker signature of interest. Now, when I worked in pharma many years ago, if I had the ability to identify a novel biomarker sequence signature for a project in a shorter space of time as I could now, I would have been really, really happy. That typically would have taken years of work with lots of validation. So this actually provides you, it's a game changer. It's a paradigm shift, which actually enables people to accelerate their research now to a level that they couldn't do before. So if you didn't want to run the whole of the 3,000 analyte panel that we have something called Explore 384. And basically that, I can explain that in the Explore 307.2, 3,000 analytes is broken down into 8 disease-focused panels. And you can basically pick one or more of those panels. So there's a cardiometabolic, 2 cardiometabolic panels. There's 2 inflammation panels, 2 oncology panels, and 2 neurology panels. So as I mentioned, you can pick one or more of those and maybe have a bit more of a focused approach. And the Explore actually covers all the major biological pathways and processes. So you don't need to analyze the whole proteome if you didn't want to. You can actually analyze Explore, and that will cover everything that's actually relevant. So moving on to what we call our mid-plex product line. As I mentioned earlier, this gives you a qPCR readout. And I'll start off with the largest, let's say, panel that we have, which is called Target 96. All of these, incidentally, you can run on our benchtop instrument, which is called the Signature. What I didn't mention, actually, is with our technology, it's accessible to anyone. And the way we make it accessible is we could actually run it for you in our labs. Partner labs can run it for you. So we have over 60 partner labs that can run it, and Source is your local partner lab here that can run those projects for you. Or you could run it in-house. So if you didn't have an NGS instrument, I advise you go to Source to run it. But potentially you could purchase a Signature instrument if you wanted to run routine assays. That might be a way forward. Or there'll be partner labs, and I'm hoping the Source probably in the future, I expect they would invest in in the midplex instrumentation in the future. So to talk a little bit more about those midplex panels, Target 96, that basically consists of, I'll say, 14 human disease focused panels and 1 mouse exploratory panel. Each of those panels consists of 92 validated assays, so you get a 92 plex. The data, as with the Explore assays, will be in a relative output, so in what we call NPX output. And again, you can mix and match. You can pick 1 or more of those panels. If you're interested in, say, immunology or inflammation, you could pick an inflammation panel and maybe an immune response panel. If it's neuroimmunology that you're interested, maybe a neuroexploratory panel and a combination of inflammation or immune response or immuno-oncology. You know, you get the picture. So taking it down to the next level, we have Target 48. So that provides a panel which is actually focused on inflammation, cytokines and chemokines. 45 analytes per assay, so a 45 plex, 40 samples per run, but you can run multiple plates. And here we start providing the ability to give you not only NPX relative data, but absolute quantitation. And I think for some customers, this is a real key, let's say, a requirement. And I would say Target 48 probably is a pretty unique product. We have other competitors out there, other immunoassay platforms. I don't think there's anything that really can compete with Target 48 that will allow you to run so many analytes as a single plex, give you quantitative data. Basically, you can use the same dilution factor, etc., for them. With the other panel, say competitor assays, you're probably going to have to run multiple panels, which is going to take extra time, cost, and utilization of sample. So the next product I'm going to talk to you about is Olink Flex. So this product was released in February this year. And what this now offers you fills the gap for those that want to do maybe more specific, smaller plexes. So it will allow you to run between 15 to 21 assays. And here you get to choose which analytes you want in that panel. So we have a panel of around 200 analytes at the moment. They're inflammation focused. We will be bringing out other panels in the future. You pick, which assays you want to make up between a 15 to 21 plex from that list. You can get the data again in absolute quantitation, and we'll basically ship the reagents to you or your partner lab within 3 to 5 weeks. So it gives you full flexibility. If you want further flexibility, we have a product called Focus. So the example of when you might want to use this is if you've run an Explore project, you've identified a novel signature that you're interested in, and then you want to basically run assays with that signature routinely. So you could use Flex if the assays you were interested were on that Flex panel, but if they weren't inflammation assays, analytes, and maybe covered a broader range, we could develop a specific focus panel for you. So anything between a 1 to a 21 plex. And again, that would be in absolute quantitation. It's a longer term project. It will take several months to develop that for you, around 6 months, and there'd obviously be a cost associated with that. So you have a complete, let's say, portfolio, which actually provides you full flexibility. It provides you the ability to go from exploratory or discovery phase right through to, let's say, lab developed test, LDT, or even IVD. So our product line is actually only for research use only, but we have examples of people who are using it as an LDT, where they do the validation in-house. And also we have customers that are using it in the diagnostic capacity, but you would have to basically do that work yourself. But we would support you in your ability to do that. So if anyone is more interested in that sort of workflow, we can have a discussion about that later. So I won't go into too much detail, just to talk a little bit about the validation. So we validate our assays as you would expect for any quality immunoassay, but we also do this for our Explore product. So we'd look at sensitivity, we'd look at precision, we'd look at scalability, i.e. does the data, a single plex look the same as it does in a higher plex assay. We look at specificity and also the dynamic range. In addition to that aspect, the quality aspect, Olink is a really supportive and collaborative customer. So those of you that have worked with us will know that. I think it differentiates us from a lot of, let's say, other companies. We have the ability to do everything for you, from basically helping you with a design study. We have field applications team that can do that. We also have a sales team like myself, which are scientists that are trained and have experience maybe in certain fields. Some are experienced in assay development and working in pharma, preclinical validation like myself. Some have population health skills. Some people work more on the genomic side of things. So we have people that are highly skilled. Then in terms of technical skills, we have the field application team. And then we have people with even further depth in terms of scientific skills, such as Mariana, that know the literature for specific areas. They have specific, say, research areas of specialism, and they know the scientific community as well. So they work very closely with a lot of the KOLs, so they can put you in contact with the right people, all the right kind of collaborations and consortia that we've set up. And then we have data analysis team. So data science, they're very let's say, experienced in that side of things, which I'm not, and we can put you in contact with those. And then we have a production team as well, manufacturing team. So we have, say, a company around 700 individuals, but we're all focused on this one technology and very efficient and very good at what we do. And we can handhold you, or we can let you walk and run at your own pace, depending, wherever, what stage you're at. We can help you do that. So I think it's my final slide. So in terms of Olink, we can provide a quality product with high sensitivity, high specificity. It's tried and tested. So there's over 1,300 publications, and you have a look at the publication list, you'll recognize some of those key players that are using it to give you some reassurance of the quality. It's reliable platform. We have over 62 partner labs that run it routinely. The data should be, as good quality between all of those labs. There's a procedure that we actually have to basically train those labs and make sure they're ready to run before they do. And then the technology is scalable, can take you through from discovery all the way through to, let's say, diagnostic level.
Yes, it's a pleasure being here with you today. So thank you so much for coming. So today I'll share with you how Olink Proteomics can really help advance biomarker discovery, information research, multi-omic approaches.
You should be talking to the mic.
Oh, that's unfortunately I can't extend. How can I?
Oh, maybe we just need to
I
get
think we
it.
need to connect, perhaps.
Should be fine. She says. Oh, there we go. Oops, sorry.
Oh.
Should be fine now. Yeah, there we go. It's
Sorry.
just a bit stuck.
Oh, that's all right. So omic approaches have really shown the promise and the ability to accelerate drug development and the drive towards precision medicine. And indeed, GWAS studies have uncovered really countless links between genetic variation and disease biology. We also know for several years now that clinical programs with genetic data are twice as likely to be successful. On more recent news, protein-based endpoint biomarkers have shown to provide a faster route to market, as exemplified for an ALS program using the levels of neurofilament light. And also clinical trials that are leveraging stratification markers have shown to gain very significant clinical efficiencies by being able to require up to 80% less patients to be enrolled. So really highlighting a few examples of how omic approaches are really adding value to the space. However, since 2015, what perhaps you experience, and we certainly observe, is a tremendous growth in the interest in integrating all the different omic approaches to add value to clinical research. And this is because it allows us to have a better understanding and more complete picture of human biology. And that's exactly how we see proteomics as one additional layer of important information that could be added and should be added to valuable insights from other omic approaches. And as Richard mentioned, we do focus on proteins. And the reason why we do so is because proteins have a real-time cellular function. So we know that proteins are driving, and they're also the factor of several cellular functions and biological processes. And the proteome is also very dynamic. So it changes with the environment, with pathological conditions, and therefore is a great proxy for or a great insight into real-time biology. And finally, also, the vast majority of drugs are actually targeting proteins. So it's very important for us to be able to have a good understanding of the proteome. And when Richard already touched on this, but I just want to highlight this point, that when we look at matrices such as serum and plasma, which are very often studied because they are minimally invasive and constitute a very good source for biomarkers, we actually looked at 99% of the protein content in these matrices can be attributed to just 22 highly abundant proteins. So what this means is that only 1% of the protein content in these matrices, you have all the low and medium abundant proteins, which I know are very important for biological functions and can be a great source of biomarkers. So it's really not surprising that in traditional proteomics, we haven't been able to really target the entire proteome. And that's why I really just want to highlight the advantage of Olink is really be able to uncover this broader dynamic range and give information also about the low and medium abundant proteins. And this led to really brilliant contributions, which we've seen from our customers and partners across a broad range of therapeutic areas with more than 1,300 publications, and definitely fast growing in the last few years. And these are very important to advance scientific discoveries, but also to build that body of evidence for clinical programs. So I'd like to start by sharing with you a few examples of how the technology is really enabling biomarker discovery and give a sneak peek into different cases. So looking at disease classification, disease progression, and also treatment response. So for me, being in the neurology field primarily, I find this study really, really important because it's a study that's looking into profiling the CSF proteome of patients in the Alzheimer's disease spectrum. So keep in mind, this is a very complex disease with a very long latent stage, and also that the disease in itself, Alzheimer's disease, has a very strong overlap with other diseases that lead to dementia, such as dementia with Lewy body and frontotemporal dementia. So it's really difficult to diagnose these patients appropriately. So this group from Amsterdam UMC did this large discovery study. Thank you. Did this large discovery study where they looked at the CSF proteome of controls, MCI, so mild cognitive impairment, so early stages of Alzheimer's disease, and also patients with actual Alzheimer's disease. And they also looked at patients that have other non-Alzheimer's disease dementia cases. And they were able to find a large number of differentially expressed proteins in the different groups. And they built these classification models, which allowed them to differentiate Alzheimer's disease patients from controls and Alzheimer's disease patients from non-Alzheimer's disease dementia patients. So they started with this discovery project looking at 14 target 96 panels. And then they actually built 2 focus panels, which Richard mentioned. So these are the custom panels with just 8 analytes to differentiate between AD and controls, and 9 analytes to differentiate between AD and non-AD dementia. And what they found in these curves that you can see in the panels on the bottom is very strong classification power accuracy. So as you can see here with the areas under the curve of 0.96 for AD versus control and 0.87 for AD versus non-AD dementia. So a strong ability to classify disease and also to differentially diagnose. And this is an example of a company, Octave Bioscience, which took it a step further towards clinical implementation. So in this case, they're really supporting and driving the space of precision care in multiple sclerosis. And they focus on building a test that could accurately give you information about multiple sclerosis disease activity and progression. So again, they started with this workflow of doing a large explorative study, but then built, as in the previous group, they built a focus panel to look at just a subset of proteins, 21 proteins, but then they took it a step forward and did analytical validation in large external cohorts. And they've done a lot of also preclinical or pre-analytical validation to ensure that their assay could be used to support clinical decision-making. And so as of October last year, Octave was able to launch their LDT, laboratory developed test in the US to support multiple sclerosis disease activity assessment. And finally, I want to give you an example of looking at biomarkers to identify novel biomarkers to predict immunotherapy effectiveness. So immune checkpoint blockade drugs have really known to be adding a lot of benefit and really driving the space of oncology therapeutics. However, only in 30 to 35% of advanced cancer cases, they are actually effective. So it's really important to identify strong biomarkers that can help us understand which patients would benefit from this approach. So this group, based at INSERM in France, they've done a large, so they did an Olink Explore 1500, so looking at around 1500 proteins in a set of a pan-cancer group, mostly looking at lung cancer patients. And they look at these patients before and after treatment, and then they also validated their results in a larger validation cohort. And what they were able to identify, and hopefully can recognize in the graphic on the right, is that one of the proteins that came up in their analysis, which is called leukemia inhibition factor, or LIF, is really a strong and potential biomarker for immunotherapy resistance. So what you can see in this graphic, where you have in the y-axis the overall survival percentage, and over time, its progression on the x-axis, you can see that patients that have a low LIF levels, they progress, and they have an overall survival, a higher overall survival. While patients that have high LIF levels, they tend to progress much quicker. And so LIF levels are really a poor prognosis for overall survival. Importantly, they also looked and correlate LIF levels with other multi-omic approaches, looking at the tumor microenvironment. So they looked at both RNA-seq and multiplex IHC, and they were able to correlate these levels with clinical ecological features. So here I showed you around biomarker discovery studies, but I also want to give you an example specifically within this pipeline of drug development of how the technology is being used. So I'll give you a few, or just a glimpse of a few examples of how the technology is being used throughout this process. So starting with identifying new drug targets. So these would be large explorative studies. And for that, as Richard mentioned, we have a few consortia, and there's some beautiful results around identifying new drug targets that came through the, through these approaches. So Scallop is really one of the pioneer efforts to develop the field of proteogenomics. So where you combine information from genomics with proteomics to identify important biological insights. And this collaborative framework is led by Dr. Anders Malersik, who has a dual appointment between Pfizer and the Karolinska Institute. And what you can see is that through this initiative, they were able to bring together 35 investigators and bring together and harmonize the data from 45 cohorts, which represents around 70,000 patients and controls. So really powerful information if you want to do for example, meta-analysis. And one of their milestones, really publications, is they look at this. They did this meta-analysis at looking at just 90 proteins. This is a target 96 to look at cardiovascular proteins. So just a small assay, but across a large number of samples, so around 31,000 individuals. And they were able to identify very significant PQTLs. So these are very robust associations between genetic variants and circulating protein levels. And with this approach, they were able to validate previously established or previously identified targets in other studies for cardiovascular disease. They're able to also identify novel targets, protein targets, and finally also identify and uncover very interesting repurposing. Actually, we've heard recently from some of the individuals in this consortium that they are now in the process of validating some of these potential hits, especially for the repurposing field. So that's quite an exciting way because, of course, it drives the process of development much faster if you can look for these repurposing opportunities. And then a project, Ali, I think you'll appreciate seeing, one of the projects that really took on the steps from Scallop is the pharmacoproteomics project. And this is really a remarkable initiative where 13 pharmaceutical companies came together to commission the proteomics profiling of more than 57,000 patients, or not patients, participants of one of the most respected and really well-characterized population scale biobank, which is UK Biobank. And with this approach, they run our Explore 3072 in this large number of individuals. And with that approach, you're able to unveil the largest PQTL collection to date. And this is very exciting because this resource it will be or is already being made open access. So everyone can access this information, just need to make the request and explain the reason why you'd like to access this information for the purpose. But really the goal of the consortium is to accelerate the development of novel biomarkers and to elucidate biological mechanisms that are underlying on these genetic discoveries. So the first part of the data was released in April this year. So now you have access to proteomics data for 1,500 proteins, so just half of the entire profiling in 57,000 individuals. And of course, these individuals also have genomic and clinical data available. So this, I hope that I was able to share with you some examples of these large studies that can give information about novel drug targets using genomic approaches. But I also wanted to touch on the piece around preclinical research, where you might not really be looking at plasma or serum, but you might be looking at other matrices, and you might want to be doing some functional, more targeted approach. And I want to show you that it's also possible to do so using Olink. So in terms of the compatibility, we mostly speak about plasma and serum because that's how we do our extensive validation in-house. But actually, the platform has been used, and there's many publications across a wide spectrum of matrices, and we know it's very compatible across these matrices. So I want to give you just an example, again, a bit biased on the neurology side, but it's an example of a matrices which is quite complex, high fat, and perhaps sometimes you get difficult results from it. But you can see that, for example, if you look at brain tissue, you can get quite good detectability using Olink. So this is looking at Olink with Explore 3072, and these are some of the key markers, inflammation markers, and you can see that in brain tissue, the levels are very significantly above the limits of detection, and you across all panels, vast majority of panels except for oncology 2, the results are above 70%, which is very good, especially if you correlate with RNA-seq. That's what you would expect in this matrix. But I also want to give you an example on iPSC derived cells, as this is a matrix which is, as I believe some of you here will be very aware of. There's tremendous interest in the community in this space, and we see more and more publications and interest coming through, and the technology has been used very successfully in this matrix. So this is an example that was done, a collaboration between the University of Gothenburg and AstraZeneca, where they did a characterization of a human stem cell model of cardiac hypertrophy as a model to later on looking at heart failure. And what they did is they looked at iPSC cardiomyocytes which they differentiated, and they induced those with endothelin 1 in order to be able to recapitulate cardiac hypertrophy. And then what they did was a strong characterization of the cells. They lysed the cells and they run those by RNA-seq, and they collected the secretome of the supernatant of the cells, and they ran them by Olink. And using this information, they were able to actually identify 23 relevant differentially expressed proteins, which also had a strong correlation with RNA-seq data. Interestingly, and I think you would expect that, they did see a time delay between the cells and the expression levels. But I think you would recognize that a 0.8 correlation is quite a good correlation if you're looking at also considering that there's some intracellular proteins which will not be secreted. And using this approach, they built the middle model, which allowed them to identify multimodal biomarkers for cardiac hypertrophy progression, which was their ultimate goal in this approach. We can go in more detail, perhaps in the discussion, if anyone has questions on how to use these matrices. I just want to touch also on the clinical trial side, how exactly the technology, just give you a few examples of how the technology is being used already in clinical trials. So there's actually more than 50 clinical trials they are using or have used Olink so far in different stages of the process. And also, as you can see, in different therapeutic areas. And this is data that you can easily access. If you just go on clinicaltrials.gov and you search Olink as a term, you can find and you can extract all the trials they're using. So I want to give you an example on a phase 1. So this is an example in the space. So for a therapeutic program, looking at immunotherapy for ALS, and what they found is they found that in this trial that the treatment was safe and tolerable in these patients. However, the degree of the progression was very different between this small group of patients. So they found within the treatment group, they found 6 patients that had a good response, so they didn't progress after the treatment, but they had 2 patients that progressed very quickly, so they didn't respond to the treatment. And when they looked into the proteomic analysis, they identified 2 proteins, IL-17C and IL-17F, which the levels were not only high from the beginning, but actually increased even further as the disease progressed throughout the treatment approach. So they took this information as an interesting insight that perhaps they can incorporate before enrolling patients into the trial, as if they have high levels of these proteins, they are perhaps less likely to respond properly to the therapy. So this is a second, this is a phase 2 trial, and in here, so in here they were testing the effectiveness of a combined metabolic activated approach to treat Alzheimer's disease. And this is because we know that in Alzheimer's disease, there are several processes associated with the mitochondrial function, and just bioenergetics, which are disrupted in these patients. So what they did in the study is, so they had placebo and the CMA treated group, and they took samples at different stages, and they compared after 84 days with the baseline at day 0. And interestingly, I want to give you an example of the different type of multi-omic approach that have been used. Here, they looked at clinical parameters, so a lot of cognitive testing assessment and imaging assessment, but also looked at metabolomics and proteomics using Olink. And what they were able to find, and you can see here on the panels below, is they were able to validate the effectiveness of their treatment. So they saw that the plasma levels of proteins and metabolites are associated with NAD+ and glutathione metabolism were significantly improved after treatment. So they effectively, the treatment worked, and they could have this information to look at target engagement. And finally, this is my last case. I want to show an example of how you could use Olink, even if you already have a drug out there in the market that is approved for different indications. So this is the example for empagliflozin, which, sorry, it's a bit of a busy slide, but which in 2014 was approved for type 2 diabetes, and then was further expanded in 2016 for type 2 diabetes with cardiovascular disease, and finally, more recently, actually generalized for use in cardiovascular disease patients. And so it really shows the evolution of this drug and the application to the broader patient group. So what I wanted to show you here is this study. This was a really large study which aimed at further understanding how this drug is impacting and what is their mode of action in to improve the levels of heart failure and renal function. So they did Olink Explore on more than 1,000 patients, and they did Olink Proteomics at 3 different time points. So at baseline, at week 12, and week 52 post-treatment. And these are all patients with heart failure, different degrees of heart failure. And they were able to identify several processes that are affected in this disease, and they were able to identify specific molecules that are differentially altered in this condition, including those associated with autophagy, inflammation, fibrosis, and cellular stress. So these allowed them to get further insights and further evidence about the mode of action, and hopefully even further help with the expansion of their label. So which supports can you leverage to support your results, but also to get started? Olink Insights, as Richard mentioned, I think this is a great resource to get started. If you found that it's interesting, there's some potential applications, I would suggest to look here. Celine was asking from the Finegen, she was asking around which specific targets can I find, or people are also asking what is the coverage that you have, Ali was asking. So we have this password browser where you can literally, so on the left top panel, you can go there and you can specifically say, I'm interested in hepatocyte function. And then it will light up all the proteins involved in this pathway, and then you can click further and see whether they're involved in any of the panels. So what is our coverage for those particular analytes? So it's a great way of getting started and see if our technology could be a good fit for this. And then very importantly, we've also been increasing the resources that we have here. So a very important one, in my opinion, is about the normal ranges for proteins. So looking at proteomic variability between individuals. So you have an idea in your plasma, what are the levels that you'd be expecting? Or if you're using a different matrix, what are the levels that you'd be expecting to find? And very importantly, here you have data around across the 3,000 assays, across 3 different cohorts, international cohorts from 3 different continents. So China Kabur and also the UK Biobank and also Darival from the US. So you have good diversity in terms of the individuals that are included in this cohort. And finally, on the disease atlas, you have information for 15 different types of cancers where we actually have data available and information available about their profiles. So hopefully that gives you some interesting information to get started. And if you're already have some data, but you want to further explore and work with other researchers, we do have several consortia which are open opportunities to discuss with others, challenges, but also to share data, share access to different cohorts. And this is just one example. So it is the coral, which looks at neurological disease and had by Professor Charlotte Tennyson, but we also have for IBD, and as I mentioned, Scallop and growing number of consortia, which are a great platform for scientific discussions. So this is all I had to share with you, but looking forward to the discussion and help you get a better understanding of human biology.
Yes, I'm Diane. I work at PlaqueTec, which is based here on the Baberham Research Campus. And I'd just like to talk to you about the way that PlaqueTec has used Olink Proteomics. And we've used it to analyse coronary trans-plaque gradients. And this is a novel approach to understanding coronary disease biology. So I'll just take you through why we would actually want to analyse trans-plaque coronary protein gradients. Well, you may be aware that there is a huge unmet need and a lack of precision medicine in coronary artery disease, or CAD. And although there are really long established population level risk factor optimization, we know that that works. However, there is still a residual risk that clearly remains. And that's shown here in some of these numbers that are taken from the World Health Organization. So it's reported in 2019 that every year, 17.9 million people die from cardiovascular disease, and this accounts for 31% of all deaths. And that 1 in 3 of these are actually people under the age of 70. And in another study I've referred to here, it reports that 6% of Americans are living with coronary artery disease, with 1 heart attack every 42 seconds. So this is an incredibly prevalent problem. So there are currently precision medicine approaches by other people, and we've heard about some approaches even this morning in discussions. And typically what tends to be used is a systemic blood sample to look at various parameters in the blood. And there are actually some biomarkers of disease, such as CRP and some others, which actually do act as quite good predictors of risk of coronary disease. But there have been no real major breakthroughs in drug development. And also what the systemic blood sampling does is it does have these potential risk indicators, but it doesn't actually tell you where if there's an increase in a particular protein in the blood, it doesn't tell you where it's coming from. So it's not specific to coronary blood vessels. So the approach at PlaqueTec is we have this unique blood sampling which occurs at the site of coronary disease. And we're using this in order to understand the underlying biology. And our ultimate aim is to try and answer this question at the top here, is to identify endotypes. And that's to really be able to place patients into a particular subgroup based on their underlying biology. So how do we actually do that? Well, I've outlined here, this is basically an overview of how we use the PlaqueTec device, which is also called the LBS, or the liquid biopsy system. So we collect coronary blood, obviously from the heart, and we also have some reference blood samples taken as standard venous samples. And this is a cartoon image of a coronary artery, a section through a coronary artery. And I'm indicating here that the blood flow would be in this direction, from left to right, or from upstream to downstream of the blood vessel. Here in yellow, we have an image of an atherosclerotic plaque. And the sort of dots that you can see here represent some of the biomolecules that may be released from the plaque and accumulating along the boundary layer. So the device here, the LBS shown in blue, is inserted into the coronary artery in a live patient who is going in to have a coronary procedure. And it's inserted before they have one of these procedures. And you can see here that the baffles that are on the device, what these do is they ensure that turbulence occurs. So there's a mixing of the blood, which allows this efficient collection of biomolecules that might be accumulating along slower moving boundary layer on the surface of the endothelial cells of the glycocalyx. And we're able to collect blood samples simultaneously at multiple points along the coronary artery. And you can see here, these are indicated in red. So there's one downstream, one upstream, upstream here, and a midstream sample. And by comparing the downstream samples with the upstream samples, this can actually reveal gradients in protein concentrations, and it has the potential to detect biomolecules at elevated levels associated with plaques. And we predicted that this would help to uncover some novel insights about coronary artery disease and what the plaque might actually be releasing or leaking out from the plaque. So we have two clinical studies to date. These are quite small studies. The first one was called First in Man. It's abbreviated as FIM, and this looked at sampling from a healthy blood vessel, first of all. And the second study was called CS1, Clinical Study 1, which actually analysed patients with symptomatic plaques. And we analysed blood samples from the device using Olink Proteomics. So these first experiments happened back in 2015, quite some time ago. And I think we were probably one of the first users of the Olink platform. And why was Olink chosen then? Well, the samples that we have from the coronary artery are small volume. They're very precious samples. And the Olink assay required only 1 microliter per sample, as you've heard this morning. The panel that was chosen to look at, first of all, was called the Cardiovascular 1 or CVD1 panel. It's now discontinued, but this contained 92 proteins, which covered many of the proteins of interest to PlaqueTec at that time. And we had evidence that even back then, there was high precision and high sensitivity, which is what we required in order to be able to compare the samples that we were interested in. So this is these are results from that first study. This publication came out at the end of 2017, and I won't go into huge detail about this, but I would just like to point out that what we have here are 3 volcano plots, and on the x-axis we have the difference from the downstream to the upstream sample and the p-value on the y-axis here. And what you can see in 10 patients that were sampled, the 10 healthy patients from the First in Man study, is we saw some proteins released. Each of these dots here, by the way, represents 1 single protein. We saw some proteins being detected, but none that were significantly different downstream compared with upstream. In patients which did have symptoms, this was 12 patients that were sampled prior to the procedure, we found there were several significantly different proteins at the downstream compared to the upstream site. And in 27 patients who were sampled after the coronary procedure, and typically it was a stent that they had put in which disrupted the plaque, you could see that not only were there significant proteins released, but there were additional proteins released. And the take-home message from this slide really is that what we saw here was statistically significant mean protein gradients that were in common for all the patients in the whole cohort. But how about the individual patient profiles? What did they look like, and how does this relate to peripheral levels? So I've been looking at some of that data more recently, and I thought it might be helpful to actually highlight the kind of data that we have at PlaqueTec by just actually focusing on one protein and looking in the 12 patients that had symptoms and were sampled prior to the procedure. So the one protein that I've chosen, I've coded it here as V1, and we're starting off just looking at the peripheral samples only. And what we have is 12 patient profiles here from the CS1 trial. And what you can see, so what we have is on the x-axis, we have the individual patients and their ID. And on the y-axis, we have the absolute value or the NPX value for this particular protein. And I think what you can notice is that patient 9 stands out. They have much higher levels than the other patients. Then when we introduce just one extra coronary sample, so this is looking at a coronary upstream sample. And what you can see with that single extra sampling shown in blue here is that the sampling from just one upstream sample, so this is prior to the disease site, it pretty much reflects broadly the systemic levels. So there's a fairly good correlation, and still patient 9 stands out as having the highest levels of this protein. But then when we introduce the downstream sample, shown in red here for these patients, these downstream samples, whoops, what have I done? Try to hide the really interesting observation. Okay. So what we have here is that we don't see a mirroring of the upstream or the peripheral samples for some of the patients. And I'll highlight this just by pointing out patient 7 here, just to give a good example, that where we've got relatively low systemic and upstream coronary levels of this protein, we actually see much higher levels of it in the downstream sample. And what this suggests is that there are higher levels of this protein associated with where the site of the plaque, and this protein might actually be released or leaking from the plaque. So this tells us that, for example, if a drug company were targeting this particular protein with the systemic data that anyone can get access to, that they may have focused their activity on patients like patient 9, where they might want to reduce its levels. Whereas from the analysis that we're doing at PlaqueTec, it would probably make sense to include patient 7, also patient 10, similar pattern, and definitely also patient 43. So we're able to see a lot more about each individual patient's protein profile.
So
Yeah.
the pattern is very similar on patient.
The pattern of the pattern that you see is similar. I think if you are looking at the systemic sample and the upstream coronary sample, but with the downstream sample in red here, we can see that there's quite a difference between the upstream and downstream sample, which I think tells us that there's something else going on within the coronary artery for that patient. And also for this patient here and here, not for these patients, they don't seem to have much difference in within the coronary artery. Is that clear, or?
Yeah, I was thinking it seems that all of them has a downstream higher level than
For most of them, they do have a downstream signal that's higher, but it's not necessarily much higher. Some of them have small differences within the coronary artery, and some of them have much larger differences.
How do you measure? How do you know actually which one is the one? I mean, obviously you see here. I mean, is there any like a standard way of saying if it's this much higher than I'm?
Well, I can take you on to the next slide, which I think, because the reason for presenting you the data in this way was just really to show you the raw data and to see, just by looking at one protein, to give an example of how we're actually capturing that extra data and getting some extra insights into what's going on within the coronary artery.
A
Yes.
quick question, because you mentioned the reason why initially you engaged Olink was because of the sensitivity of the assay.
Yeah.
So what were you guys doing before Olink?
Oh, well, they're
And could you?
looking at
say
Luminex
how
assays
big-
really,
Yeah.
before. Yeah, and we do have some comparisons with that, and they do
Oh.
seem to correlate well. But I think the precision and the sort of smaller CVs were something that was really appealing.
Okay. You're gonna.
I should also point out that I did not work at PlaqueTec when these initial experiments were done, and so that's my understanding of why Olink was chosen.
Okay.
Okay, so coming on to the next slide, this might be more informative to help with your to answer your question. So yeah, when I came to PlaqueTec, I did some repeat analysis because I wanted to see if we would how well the assay repeated, and also wanted to explore a little bit further on 2 other panels. So at this point, what we did was repeat using the same plasma from the same patients, using a cardiovascular 3 panel and also an inflammation panel. And quite a lot of the proteins on CBD3 and the inflammation panel did overlap CBD1. But what I've been able to do here with all of the 252 proteins that we were able to measure, what I've done is plotted the same 12 patients on the x-axis and plotted here the intracoronary ratio. So we're just focusing on those coronary differences in the patients here. So this is looking at downstream divided by upstream. And what we could see, which was interesting, is the majority of the proteins actually had a gradient of around about 1. So actually not showing much difference between the downstream and the upstream samples. So with no change across their coronary blood vessel. However, the red dots above 1 here, these are proteins which stood out as having higher downstream compared to upstream ratios. So these red dots are representing proteins which are essentially our hits for where we're actually detecting some differences downstream compared to upstream and associated with that person's atherosclerotic plaque. So another thing to highlight from this graph is that, some patients, like patient 53 here, has very little activity within their coronary vessels, whereas, for example, patient 10 here has a lot of activity, so does patient 7. So this allows us to see and really highlights the interpatient variability. And what we'd really like to do next is to validate these results and also to explore even further in a new trial. And I'll just take you through in the next slide. This just outlines what we're planning to do next. So this is an analysis of patients. It's called biopattern, the trial. Analysis of patient samples. These patients have established coronary disease and they'll be undergoing an intracoronary assessment. So the clinical trial is going to be based at 8 different UK sites and it's managed by Papworth Clinical Trials Unit. And we're aiming to start in autumn this year. There will be 300 patients. There will actually have 4 coronary samples taken from each patient. And each patient will also have 3 peripheral samples. We'll have 1 baseline venous, a systemic arterial pre-heparin sample, and a systemic arterial post-heparin sample. And the reason for having these is to have additional controls for all of the coronary samples. So we'll have both coronary and peripheral plasma. And those plasma proteins, again, we're going to use the Olink Explore platform and look at 3072 proteins, really so that we can explore as much as possible with the samples and also validate our earlier results, because this panel does include our earlier hits. And we also plan, depending on how much plasma we have left over, we would very much like to look at other biomolecules such as lipids and metabolites. And then with the cell pellet remaining from the plasma isolation, to hopefully do some cell phenotyping and to also look at RNA and DNA. What we're planning to do is to, again, look at analysing biomolecule gradients in the coronary artery, and then to take that data and to integrate it with looking at common SNPs that the patients might have. We'll also have some extensive imaging data, including optical coherence tomography, also called OCT. So we'll be able to get an image of everyone's who takes part in the trial. We'll get detailed imaging of what size and type of plaque that they have, and we'll be able to correlate that with all of the other biomolecule data that we have. With extensive patient demographic data, and because the amount of data we'll be generating is going to be very large, there'll be multiple blood samples per patient, 3,072 proteins, and all of this other data means that we will be needing to use some machine learning approaches in order to pick out some of these relationships. So the value of the results, we're expecting to be able to endotype coronary artery disease, and we think this will provide new insights for coronary disease biology, also for biomarker discovery, to be able to find signatures for particular types of coronary plaques. And of course, we expect this to lead to new drug targets. And actually, one of the targets, one of our hits from CS1, we're actually going ahead with a drug screen, repurposing screen already. Obviously, we hope that this one will be validated in the new trial, but this is something that we're doing in collaboration with one of the academic groups here at the Babraham Institute, and also with a company based here on site. So, so again, for this new trial, why are we using Olink? I thought it was important to mention that for this particular meeting. So again, the sample volume is small from the coronary arteries. These are really precious samples. The assay for 3,072 proteins requires 80 microliters per sample. Again, it's high precision, high sensitivity. I think it's trustworthy. We've shown some in-house reproducibility and good correlation with standard ELISA tests. I've found that any questions we have about our planning, the technical support has been fantastic and really, really helpful. The only limitations I can think of have already been mentioned this morning, that although there's a really wide coverage of the proteome, it is a pre-selected and somewhat biased list, and that what we get is protein levels and not necessarily protein activity. We don't know about any post-translational modifications that might be on the proteins that might be important. And lastly, as mentioned, cost is a limitation. It is quite an expensive technology. However, I do think that if we are able to get these unique insights, then it's very valuable. And if we have time, I'm not going to go into this until this is my last slide. And it was just really to highlight something extra that we found from the initial analysis at during that CS1 trial. So this was a surprise finding, which I thought was worth mentioning here, and because it was using Olink Proteomics. If we just focus back on the CBD1 panel here, what I did was compared the coronary levels with peripheral plasma protein levels, and found this really quite striking pattern. And what we have, so it's the coronary protein levels divided by the peripheral levels. So we've got this ratio here, and you can see that some of these numbers are quite high. These are quite big numbers. And what you can see is that for hepatocyte growth factor, or HGF, campylysine 1 here, these really stand out as having much higher levels within the coronary artery compared to the periphery, but for 10 of the 12 patients, not for every single patient. And so there's 2 patients down here where it looked like there was no change. And I've also highlighted SPON1 here because it had that same pattern of 10 patients having higher levels in the coronary compared to the periphery. And because that's a ratio that we're looking at here, I've actually just taken the example of HGF and showing you the raw data here, the NPX values. And this is explained now by the coronary levels actually being very high. Every single patient has high levels of HGF. And when you look at the peripheral levels, there's 10 patients that have very low levels of HGF, whereas these 2 stand out as having levels similar to the coronary levels. And this was quite an intriguing pattern, and it was very similar for SPON1 and papalysin1. And we've actually come up with this signature definition, which is a combination of HGF, papalysin1 and SPON1, all in above the 90th centile, actually helps to describe this pattern. And when we started to see that we could see the pattern in other cohorts in freely available data, we thought, okay, there might be something in this. And in collaboration with a group at the Karolinska Institute, we were able to find out that in patients that had, or not patients, actually participants that had more than 3 risk factors for coronary vascular disease, in that particular cohort, there was outcome data, and we were able to see that individuals who had the signature pattern actually had a 3-fold increase in coronary events compared to those that didn't have this signature. And also, and again, this is freely available data, the MGH Boston COVID-19 cohort, we were also able to detect participants with this pattern and that they had higher incidence of coronary events and also higher mortality rates. This is all written up and published as a preprint in medRxiv. It came out recently. And what we'd love to do is to look in more cohorts to see just how relevant this particular signature is. And our plan is to access the UK Biobank study, which was mentioned in the last talk. And so by looking in lots of other cohorts, we'll be able to work out if this really is something that could be a diagnostic that could help identify individuals at higher risk of poor clinical outcomes. So just like to finish by summing up. So PlaqueTec is endotyping coronary artery disease, and we're doing this through efficient blood sampling at the disease site in the coronary artery, and we're building a database of coronary multi-omic data. Our pilot data suggests that these trans-plaque coronary gradients do exist, and that patients have varied gradient patterns. Our next trial will actually validate these gradient proteins and explore further proteins and other multi-omic parameters, and all of what we're doing is to enable endotyping and precision medicine for better outcomes for patients with coronary artery disease. So thank you very much for listening.